showing the variation within the clusters. And you can see there's kind of a small peak there, but it exists for the bdelloids. There's a much larger peak in the monogonons. So as a result of this, the species pattern of clustering is much stronger in the monogonons. And the conclusion from this is slightly counterintuitive. The species are more distinct in monogonons. That's because the bdelloids appear to diversify more than the monogonons. In the same period of time, a bdelloid clade has diversified into 3 times as many clusters as an equivalent clade would have done. So this would be consistent with, if you remember at the beginning I said it could be that speciation might be easier for asexuals because they don't have to acquire reproductive isolation, or it could be harder for asexuals because they might not adapt to new environments as quickly. In this case, this seems to be supporting that speciation is actually unconstrained, or it's easier in the asexuals, maybe because they don't need reproductive isolation. So they've diversified more, but as a result of that, the distinctiveness of those species is less. Now, of course, there could be other differences between the 2 groups of rotifers that also affect these patterns, and their dispersal modes do differ, actually. And we're limited here because we've only got one comparison between the monogonons and the bdelloids. But anyway, there's a suggestion there that there may be some quantitative differences between the sexual and the asexual clade, but qualitatively they're both diversifying into genetic clusters that are broadly similar. Okay. So I'm going to shift now to, so that's the speciation story is kind of done for now. I'm going to shift to the question of genetic tricks, and particularly talk about work we've been doing looking at the possible consequences of the horizontal transfer for how bdelloids evolve. So this was the first paper that reported this phenomenon by Gladyshev et al. So they were actually looking for transposable elements, I think, but by accident they found lots of genes that they identified as being acquired from bacteria and so on. This was done with long-read sequencing, so these genes are definitely within the rotifer genome. They're not contaminations. Since then, this was work by Kayara that we were involved in as well, that used transcriptome data and estimated that about 10% of the expressed genes within the rotifer had been acquired from non-metazoa. So the methods we were using to detect the horizontal transfer here were quite broad, so we weren't able to detect influx from metazoa. That probably happens as well, but we were looking for genes that were blasting matching more closely to non-metazoan genes that had been found in the rotifers here. And this value of 10% was confirmed as well in the first whole genome sequence done for rotifers by Flo et al. And Karine van Donnink's group, where they found again 10% of the protein coding genes looked like they were non-metazoan in origin. So this was quite exciting and sort of came out of the blue. And it led to a few hypotheses about what might be going on. So the first one, in terms of mechanism, then there was a hypothesis that maybe this is to do, or somehow results from the ability of bdelloids to survive desiccation and recover from desiccation that I mentioned at the beginning. So desiccation breaks membranes, and it also breaks DNA. And bdelloids somehow, we still don't know exactly how, they're able to recover from that and repair their DNA. So people zap them with gamma radiation, it shatters their DNA, and then they track them over a period of is it hours or days, and they rebuild the DNA back into pieces, and they seem perfectly happy with that. Back into, longer chromosomes. So there's a hypothesis that maybe this is behind, so you're allowing DNA into the cell, and maybe sometimes when you're repairing breaks in the DNA, some foreign piece of DNA gets incorporated by mistake. And then it's tempting to think, could this be involved in bdelloid adaptation and divergence? And so in particular, if you compare with what happens in bacteria, then it's well known that there's quite high levels of horizontal transfer. Those genes come in and allow bacteria to adapt to new habitats and so on. So we set out to investigate then whether it might be involved in adaptation, but also divergence between the species. So is the differential uptake of different horizontally acquired genes, is that a way that species can differentiate genetically in bdelloids? So this is the Silwood Park campus where I work. There's a few buildings as well, but the reason we're there is this is a field station for terrestrial ecology. So it's very similar to here, although instead of having the sea next door, we have this next door. And so this allows us to collect a lot of species of bdelloid rotifer, actually. And so we focus on 4 species of the same genus Rotaria that I've already talked about in the previous example. So there's this one, Rotaria tardigrada, is found in temporary ponds. So this one can survive desiccation. There's this one, is Rotaria sordida. So that's tends to be found on moss on tree trunks. Again, it can survive desiccation. These are a bunch of animals that were isolated from a tree at Silwood Park and then became a transcriptome shortly after this film was taken. And then we've got the 2 louse species that I already told you about. These are the ones that live on Acellus aquaticus. There's the big sister species, Rotaria magnacalcarata, and there's the little sister, Rotaria socialis. So those are 2 that were isolated from one louse from our pond in the Japanese garden. So the question then was whether has horizontal gene transfer been ongoing, and has it contributed to species differences? And in addition, because we've got, I should have said, these 2 cannot survive desiccation. They're found on a water louse that lives in water the whole time. If you try drying these out in the lab, they don't come back. So we're using this variation to test the desiccation hypothesis as well. So we'd predict if the desiccation hypothesis is true, we'd expect that there should be more horizontal transfer going on in the desiccating species than in the non-desiccating species. Okay, so Isabel sequenced transcriptomes for all 4 species and did these things. There's just some details, but there's an HGT index, so this is a measure of how closely it matches a non-metazoan versus a metazoan hit within online databases. And then we're using a particular value of that to label as foreign genes. If you use different versions of this, it doesn't change qualitatively any of the conclusions here. So the first thing we found was that I should say that the earlier finding that 10% of genes are horizontally acquired was from a different genus, Adinetta. So we found that a similar level of horizontal transfer is apparent in the Rotaria species as well. And there does seem to be a marginally higher level in the 2 desiccating species. So this is the proportion of genes that are being judged to be horizontally acquired. Note that starts at 8%. It doesn't go to 0 here. But the 2 desiccating species are marginally higher in the level than the other 2. Having said that, there's still appreciable levels in the fully aquatic species as well. So there's potentially some support that desiccation might increase the amount of horizontal transfer, but it's not completely absent in the aquatic species. So in order to look at whether this process is ongoing or not, we reconstructed the number of horizontal gene transfer events on each branch of the phylogenetic tree connecting these species. And I won't necessarily go into the details of how we did this, but it's just identifying whether there are orthologs for a particular horizontally transferred gene. Is that shared among the species, or is it unique to just one of the species? And this shows the numbers on each branch of the tree here. So these are the two louse species. And you can see Magna calcarata seems to have acquired 67 horizontally acquired genes that it has and none of the other species have. So that's evidence that there's some ongoing rate of uptake. And then tardigrada, so that's one of the desiccating species. You can see there's 303 there. The most, about half of the genes are ancient. They're shared not only between the Rotaria, but with Adinetta as well. So certainly there's a lot of this is something that's happened ancestrally in the common ancestor of bdelloid rotifers, but there's evidence from these numbers here that it's still ongoing. So that's the first result from this. We check these numbers by sequencing the genome of the Rotaria magna calcarata as well, to make sure that they sort of matched up and that we weren't missing some genes that were really there just because they hadn't been expressed when Isabel harvested the RNA, and it didn't change things too much. So. And so we could use this, because we got the phylogeny, we dated that roughly in terms of millions of years, so we could work out roughly what the rate of acquisition of these horizontally transferred genes has been over time. I think on average it was about 12 per million years. So it's not, it's a huge number compared to other animals, but it's not like they're taking them up every generation or so. And indeed, by doing this and looking at the different branches on the tree, we could see that there was a higher rate of gain and a lower rate of loss of horizontally acquired genes in the desiccating lineages. So the conclusion is that the HGT does seem to be happening less readily in the aquatic lineages, but it does still happen at an appreciable rate. So the final thing we tried to look at was whether these differences that between the species, could they be contributing to adaptation to the different habitats they're in? And this was simply classifying the different genes and what their functions might be, and it's a bit hard to go beyond that. At the moment, there's not very good tools in bdelloid rotifers for genetic modification to actually test hypotheses about what genes are doing. But there's quite a few that potentially could be involved in different dietary components. There's quite a few that are, might be related to some aspects of stress. And certainly some ancestral foreign genes that seem to be shared ancestrally among bdelloids appear to have been lost in the louse species that no longer... ancestral genes that are involved in potentially involved in desiccation have been lost then secondarily in the louse species. That was the main kind of smoking gun that we found there. But anyway, some of these things could be important for different habitats, and we'll continue to look into that. There's been a lot of press over the last few years about contamination affecting studies of horizontal transfer. There was a famous tardigrade example that reported similar levels to the bdelloid rotifers that was subsequently shown to be due to contamination, in that they sequence their genome from lab supplier cultures of the tardigrades that had loads of other stuff in as well, and the genome was, they were just assembling together all the protists and so on that were in the tardigrade sample. So we've been quite careful to check that that's not going on here. There's various ways to explore that, but in particular, as I already inferred, if you assemble the genome and you find long fragment where there's only non-metazoan genes in it, then you chuck that away, because that could be just a bit of bacterial DNA that's got taken up in your genome sequence. Sorry, I'm talking about genome sequence rather than transcriptomes now. But if you find that there's the foreign genes are in the middle of a scaffold that has lots of metazoan genes in it, then you're more confident that that's really part of the rotifer genome and that there is a bacterial gene in there. There's other evidence as well. Some of the original genes that were reported are match bacterial genes, but they've had introns appeared in them, so they look like domesticated bacterial genes. And because we're looking at transcriptomes, this 10% is actually genes that are being expressed. So the bdelloids are potentially using them, although the actual functions haven't been confirmed. These aren't sort of fragments of dead genes in the genome. So where we're at with this story is we've, moved to sequencing genomes. So we now have there's a comparison of 4 genomes here. So the original Adinetta vaga, that was a published genome from 2013, and then we've added in another Adinetta rickii, and then Rotaria macrura and Magna calcarata. So again, we've got 2 desiccating and 2 non-desiccating species here, and this I'll just show this slide here. So this is work Ruben Noel is working on. And this is showing the rotifers here and a set of other related, either phylogenetically or potentially sort of ecologically related species, just comparing aspects of the bdelloid rotifer genome with those other species. So this is the gene count, gene density. This is a measure of ploidy levels, horizontal gene content, and repeat content, so the numbers of transposable elements. And you can see the feature of the bdelloid genome that is most unusual compared to these other organisms is the content of horizontally acquired genes. So that's here. All of these other species have very few. That includes some tardigrades that desiccate, but still have very low levels of horizontal gene transfer. It's the 4 species of rotifer, some of which desiccate, some of which don't, that have high levels of horizontal transfer. So at least as a preliminary finding here, we would argue that the desiccation hypothesis doesn't seem to be what's going on and the reason for the amount of horizontal gene transfer. Maybe there's something else about bdelloids that makes them more able or more likely to take up foreign genes and for them to persist. That could be because they're asexual, but who knows? They're also quite unusual in having very few transposable elements, but there was a tardigrade here with a small genome that also had few transposable elements, so that's another interesting aspect to look at here as to what aspect of the bdelloid biology might be behind some of these unusual features. So that doesn't seem to be quite as unique for the bdelloid rotifers as the horizontally transferred genes. So the conclusion from this part is that horizontal transfer is ongoing. It does lead to differences among species. There's a slightly faster rate in desiccating species, but it's still happening in the other ones. And so we think that desiccation isn't really the key explanation for what's going on here. Now, the question that's really interesting is whether this could provide something like a genetic trick that could compensate for the lack of meiotic recombination. So the rate of acquisition of bacterial and fungal genes that we've been looking at so far is fairly slow. That's not enough to bring sort of recombination. It wouldn't have the same theoretical effects as sexual reproduction. It could be a way to acquire genes of beneficial function to enter a new niche in the way that operates in bacteria as well. But because this is happening with these foreign genes, it's tempting, or it's likely, that there will be some occasion when bdelloid rotifer genes from the same population could also get taken up by the same mechanism. And so this has led to work trying to investigate whether that's actually happening or not. So there was a paper that came out a couple of years ago by Karim van Donnink's group again, Nicholas De Bortoli, and Isabel Diego and CT, were involved in this as well, that reported evidence for genetic exchange within bdelloid populations based on looking at multiple genes and finding inconsistencies in the sort of variation between those different genes. So unfortunately, since then, Chris Wilson and Ruben and me involved as well, Chris Wilson has sort of looked at those data more closely, and it seems fairly certain now that that is actually that these apparent discrepancies are actually a result of contamination in this case. That very rarely there were multiple animals being put into the same tube, and these rotifers are very difficult to work with, then that could lead to some of these patterns. And Chris has gone through some of the sequence chromatograms and so on, and shown that it's highly likely that that's what's occurred here. That doesn't mean that this phenomenon is not happening. It just means we need to keep looking still. So this is an open question whether something similar could be providing a means for recombination among different bdelloid rotifers within populations. Okay, how am I doing for time? 5 more minutes? Yeah? Okay, so this is the shortest part now. I'll just go very quickly through some work looking more at the sort of ecology and this aspect of sex that's well known as it being proposed that sex is a mechanism for efficient natural selection, and that this would be most beneficial in trying to keep pace with parasites that you're co-evolving with. And so the big question then for this weird group, the bdelloids, is how have they managed to survive for so long in the face of pathogens? And there's a few hypotheses. So one might be that they don't really have many pathogens or parasites, that they're unusually rare, or that they're only weakly virulent. They don't have a big impact on bdelloid populations. It could be that no parasites have been able to establish a long-standing interaction. Or it could be, even if they have parasites, that they're not really taking part in the sort of reciprocal genetic interaction that is proposed in these ideas. So it's the classic idea, the Red Queen hypothesis for the maintenance of sex, that it allows you, by shuffling gene combinations, you can reduce your susceptibility to parasites that are always adapting to the most common gene combinations. So I'll just very quickly show these. This is Chris Wilson's project. He's been working on this, and quite a lot of master students at Imperial as well. So do they lack parasites? No, they don't. They have loads of really nasty fungal pathogens that attack them. So this is the remains of the body of a bdelloid rotifer here, and this is a fungal. These are fungal hyphae growing out and spores that germinate at the surface. And if you introduce this fungus to a culture of bdelloid rotifers, there's some genetic variation, but for a lot of them, it just wiped them all out. So they definitely have very virulent fungi that belong to, I mean, these are the different clades of fungi now. There's loads of them. So then how long-standing have these associations been? So here Chris has sequenced representatives. In red are the bdelloid rotifer attacking fungi. Black are other fungi. A lot of these feed on nematodes and so on. They're related fungi that were available in the database. And you can see in particular there's this clade here, Rotiferophora, which is a large clade of fungi that only attack bdelloid rotifers. And they date back to about 50 million years, which is the rough age we've got from a molecular clock for the age of the bdelloid rotifers as well. So it looks like these have been diversifying and attacking bdelloid rotifers for a very long time. So the final part was to look at whether there's genetic specificity and these reciprocal interactions between rotifers and the parasites. So along the bottom here, you've got the fungal, pathogens. Up here, you've got different bdelloid rotifer species. And then the intensity of colour here is how infectious the fungus is in that combination. And there's, I've run out of time to talk about this, which is just as well because it's quite complicated, but it's a method that allows you to partition how much of the variation is just due to the fungus, it's just due to the bdelloid rotifer, or it's through some co-evolutionary combination of those. And you can see there's a significant phylogenetic co-evolutionary interaction. So that means that related groups of fungi tend to attack related groups of bdelloids, but then another group of bdelloids won't be as affected by that fungus. So there's some matching between the genotypes of the fungus and the bdelloid rotifer. And Chris has even observed some mechanisms that might explain some of this variation, that some bdelloids are resistant to some fungi, and part of how they do that is laying down deposits around the germinating spore. So that, again, this is another area that will be interesting to look at, what the genetic basis is for this. And can we see evidence of selection on that? Is that happening in a strictly asexual clonal context, or is there something the bdelloids are able to do that is generating variation at those loci that determine resistance of the fungi? So we rejected all of these. They do seem to be somehow co-evolving with these fungi. So as I just said, that could be, maybe they're managing to do this through genetic tricks, or maybe there's some aspect of their ecology that allows them to survive this process, which could be desiccation. Maybe the fungi don't like desiccation. That's all that says. Good. Okay, so final conclusions then are bdelloid rotifers have diversified into species by adapting to different habitats. They've survived co-evolving parasites. Do they have some way for allowing recombination that we don't know about? It could be horizontal gene transfer, some mechanism equivalent to that could play some sort of role. Would that really be enough to compensate for being asexual? You'd need to look at that sort of critically to understand whether the rate at which it's happening is enough. Or is it really true that sex and recombination are not as essential as we think they are? Maybe there's some initial obstacle. The bdelloids have got past that. Now they're fine. These are all the questions we're continuing to look at. Thank you very much.
Thank you for the seminar. Questions? Be brave.
Thank you very much for the wonderful talk. I was just thinking such a high level of horizontal gene transfer paired with such a low level of transposon, which also means endogenous retroviruses and retrotransposon. So I wonder, what's your idea about the potential mechanism for the gene transfer? So is it due to what we believe can be the usual cause, that are viruses or retrotransposons, or there should be some other mechanism?
I think ahead of time I thought the desiccation hypothesis sounded reasonable, that maybe the fact that the DNA breaks and has to be repaired, and that there could be mistakes in that process, that could explain what's going on. The results we got so far don't seem to highlight that. Having said that, I mean, the non-desiccating bdelloids, they may still encounter some circumstances where their DNA breaks or where they repair it. I mean, all of this we don't know. All we're basing it on is if we dry them in the lab, they don't survive. The habitats that they're in are habitats where we don't think they will face desiccation. But it's not impossible that something, I don't know what it would be, high sunlight or something, that they might still, over the timescales we're talking about, it might still be sufficient to introduce those. I mean, an alternative might be that in the rate that they arrive in the bdelloids might be the same as in every other animal or equivalent sized animal, but for some reason they're not removed in the same way, and that could be to do with because they're asexual. Then it might be harder for them to remove for selection to remove incoming bits of DNA, or it might be that it is not as disruptive in an asexual genome as it would be in a sexual genome to have a big chunk of bacterial DNA that suddenly gets kind of spliced in somewhere. I mean, this is all speculation at the moment. I think the main hypothesis has been the desiccation one, and I think the results we have so far, they don't seem to support that, but they're not mechanistic. It's just a correlation of what's happening in the different groups, so it's probably too early to provide an alternative.
And this is very interesting. I wonder if having now the genome for some of these species,
yes,
and having been able to date, to trace the origin, the time of origin just of some of these events of gene transfer, if you could be able to find very, very young events, and just by looking in the neighboring zone on the genome for this element, you might probably be able to spot some, let's say, remnants of a potential molecular mechanism that has been driving this. I wonder if you have some results about this.
There was not by us, but there was a discovery recently of some. Can you remember what they're called, Diego? It's like super transposons or hyper mega transposons or some kind of enormous structure that seems to have replicating potential that was discovered by Irina Arkhipova. I'm sorry, I've forgotten the name. I don't think it's any of the things I just said. But they're sort of at the scale where the normal searches wouldn't have found them, and then she's discovered them. So we haven't got round to looking for them in our genomes yet, but that might be, there might be something there, and then trying to work out how those are functioning. So a lot of the classes of transposable elements, they don't have them, or very few, but they have got this, and I don't think these are unique to bdelloid, but it's some sort of new, larger scale thing that has just been discovered in them.
The Genome Organization, so in the way of chromosomes are organized, in these asexual lineages, do you see that the chromosome sizes and the number of chromosomes in closely related lineages just changes more wildly than in the sexual lineages of the neighbor group?
Well, this is an interesting question as well, actually. That is, to be honest, a lot of the focus of the genome paper we're working on at the moment is looking more at that sort of question, but it gets a bit complicated. So the view of bdelloid rotifers is that they're degenerate tetraploids, and there's a slightly complicated hypothesis, again involving desiccation, as to what's maintaining the exact structure of those copies. That you've got pairs of copies that are similar and then that are more divergent. There has been some, the first genome sequence that came out appeared to show evidence that homologous pairs of alleles were on the same chromosome, which would not really work with normal meiosis. And they were in palindrome sort of repeats, so it looked like maybe chromosomes could fold up somehow, or there were some interesting things in there. Having said that, in the genomes we've just, we're about to submit the manuscript for this. We've not been able to recover that feature in the other bdelloids that we've looked at. So that's dependent on the assembly of that original. So a year ago, I would have said there's some very unusual features that make it extremely very different from what you'd expect in sexually reproducing species. Now, I'm not so sure about that. I mean, there's species where they've counted 13 chromosomes and so on, so there's something that's lacking. The chromosomes are very hard to work with. There's a few people who visualize them But you need to do it with eggs. It's very complicated. I don't think they actually condense very often enough to do this. And so I think there is a need now for some direct chromosomal work again in the light of the genome. Nobody's done that since the first genome thing was sequenced. So that would be a way you could try to test this directly.
Other questions? Thank you very much again.